Special thanks to Thunderbird Perch for making this video possible. Nature is the ultimate innovator. It is a grand library of solutions, written in the code of life itself. Sometimes it writes the same story twice, in two different books. This is a story of such a remarkable repetition. Scientists have uncovered a fascinating secret hidden within the fungal kingdom. They found that two very different groups of mushrooms have learned to create the same powerful mind-altering compound. This compound is psilocybin, the key ingredient in so-called magic mushrooms. This discovery is not just a curiosity. It reshapes our understanding of how complex molecules come to be. It shows us that evolution, faced with similar challenges, can arrive at the same answer through entirely different means. For a long time, we thought the ability to produce psilocybin was a specialized trait. We believed it was confined to a closely related family of fungi. Their unique chemistry has been known for centuries, used in rituals, and studied by modern science. The idea was that this recipe for psilocybin was developed once, a singular stroke of evolutionary genius. The genes responsible for this chemical factory were then passed down through a single lineage, a precious family heirloom. This new finding shatters that simple picture. It tells us that nature is far more creative and resourceful than we had imagined. The discovery is akin to finding two ancient cultures on opposite sides of the world that independently invented the same complex machine. They used different materials, they used different blueprints, yet the final product does the exact same thing. In the world of mushrooms, this is a profound revelation. It tells us that the ability to synthesize psilocybin is not a historical accident. Instead, it is a trait so useful that evolution has forged it on more than one occasion. This wild convergence opens up new avenues of inquiry. It forces us to ask deeper questions about why this compound exists at all and what role it plays in the life of a mushroom. This new chapter in the story of psilocybin begins with a mushroom from a different branch of the fungal family tree. This mushroom, Inocybe corydalina, is not closely related to the classic psilocybe species. Yet it too contains the very same psychoactive molecule. The initial assumption was that the genes must have been swapped between the species somehow. This process, known as horizontal gene transfer, is like one organism borrowing a tool from another. But when researchers looked at the genetic instructions, they found no evidence of borrowing. Instead, they found a completely new set of tools. Nature, it seems, did not just copy the recipe. It wrote an entirely new one from scratch. To understand the significance of this discovery, we must first understand the molecule itself. What is psilocybin? In simple terms, it is a naturally occurring psychedelic compound. It is produced by certain species of fungi. When a living organism, like a human, ingests psilocybin, the body quickly converts it into another chemical called psilocin. It is this psilocin that is responsible for the profound effects on consciousness. Psilocin has a molecular structure that is remarkably similar to serotonin. Serotonin, a key neurotransmitter in our brains. Neurotransmitters are chemical messengers that allow our brain cells to communicate with each other, regulating everything from our mood to our perception of reality. Because psilocin looks so much like serotonin, it can fit into the same receptors in the brain. It acts like a key in a lock that was built for a different but similar key. By activating these serotonin receptors, particularly a type called the 5-HT2A receptor, psilocin can dramatically alter brain activity. This can lead to changes in perception, changes in thought, changes in emotion. Users might experience vivid visual patterns, a deep sense of connection to the world around them, profound personal insights. These effects are what have made psilocybin mushrooms a central part of spiritual and cultural practices for thousands of years across various societies around the globe. Today, psilocybin has moved from the realm of ancient ritual into the sterile environment of the modern laboratory. Researchers are exploring its potential as a groundbreaking therapeutic tool. Clinical trials are investigating its use in treating a range of challenging mental health conditions. Severe depression, anxiety related to terminal illness, post-traumatic stress disorder addiction. The preliminary results have been incredibly promising. They suggest that, when administered in a controlled and supportive setting, psilocybin can help people break free from rigid, negative patterns of thought. It seems to promote mental flexibility and emotional release in ways that conventional treatments often cannot. The potential of psilocybin has sparked a renaissance in psychedelic science. The molecule is no longer seen merely as a recreational drug, but as a potential key to unlocking the brain's own capacity for healing. Understanding how nature produces this complex molecule is therefore not just an academic exercise. It has direct implications for our ability to produce it cleanly and efficiently for medical use. The journey of this compound, from the forest floor to the frontier of neuroscience, is a testament to the powerful and often untapped chemical wisdom held within the natural world. It reminds us that the answers to some of our most pressing human problems might be found in the most unexpected of places. For many years, the biochemical pathway for psilocybin synthesis was a mystery. Scientists knew psilocybe mushrooms made psilocybin, but they did not know the precise steps. Recently, the genetic code was cracked. Researchers identified a cluster of genes in Psilocybe cubensis that work together like a well-organized factory assembly line. This cluster contains the instructions for making 4 key enzymes, nature's tiny biological machines: CSID, PSIK, PSIM, PSIH. These enzymes act in a specific, linear sequence. Their job is to take a common amino acid, L-tryptophan, and transform it through a series of chemical reactions until it becomes the final product, psilocybin. The psilocybe pathway is elegant in its directness. The first enzyme, SID, starts the process. The second, PSIK, adds a phosphate group. The third, PSIM, adds 2 methyl groups in a crucial step. Finally, the fourth enzyme, PSIH, performs the last modification to complete the molecule. It is a straight line from start to finish. For a long time, this was the only psilocybin production line known to science. The real surprise came when scientists analyzed the genetic machinery of Inocybe corydalina. They found that this mushroom also produces psilocybin, but it does not have the Psi gene cluster. It does not use the Psi enzymes. Instead, it has evolved its own, completely distinct set of enzymes to do the job. The chemical steps are different. The order of operations is different. The Inocybe factory uses a pathway that is not linear, but branched. At a certain point, the process can go in one of 2 directions, leading to 2 different but related final products. It allows the mushroom to produce not only psilocybin, but also a closely related molecule called baeocystin. Baeocystin is a psilocybin precursor, and its presence alongside psilocybin has been a long-standing chemical puzzle. The discovery of this unique branched pathway in Inocybe provides a clear and elegant explanation for how both compounds can be produced simultaneously. The mushroom essentially has a fork in its chemical road. This dual production capability is absent in the more direct psilocybe pathway. The discovery of 2 independent pathways to psilocybin is a textbook example of convergent evolution. Unrelated organisms independently evolve similar traits as they adapt to similar ecological niches. It's strong evidence for natural selection. When a trait gives a survival advantage, evolution can favor it repeatedly. This isn't shared ancestry, but arriving at the same solution twice. Nature inventing the same good idea twice. Classic example: flight. Birds, bats, insects. Same function, separate origins. Streamlined bodies evolved independently for efficient swimming. Convergence also occurs at the molecular level. Molecular convergence for a compound like psilocybin is exceptionally rare. Making psilocybin isn't a one-step reaction. It needs a whole suite of specialized enzymes. Each enzyme performs a precise chemical task in sequence. 2 different mushroom lineages evolved multi-step enzymatic pathways independently to make the exact same molecule. Unlikely, but true. That implies psilocybin production confers a real advantage to fungi. Evolutionary pressure must have driven this trait twice from scratch. This finding rules out horizontal gene transfer in this case. If Inocybe had borrowed genes from psilocybe, their machinery would match. But the enzymes and steps are very different. The smoking gun for convergence. It's a story of independent invention, not theft. This deepens the mystery and wonder of the fungal kingdom. The evolutionary toolkit is vast, more than one path to build a complex molecule. The mycelial network likely holds many more secrets. If evolution went to the trouble of inventing psilocybin twice, the logical next question is, why? What purpose does this compound serve for the mushroom itself? The fungi are not producing it for human enlightenment or to fuel new psychiatric medicine. It must give a survival advantage in their habitat. While the definitive answer is unknown, the leading idea is the antefeedant hypothesis. Psilocybin may act as a chemical defense, deterring insects and other small creatures from eating the mushroom. Psilocin, the active form, mimics serotonin. Serotonin systems aren't unique to humans. They're ancient and found across animals, including insects and other invertebrates. Those systems regulate appetite, movement, social interaction. Interfering with that signaling could scramble an insect's brain. An insect that eats psilocybin might lose its appetite, become disoriented, or be unable to keep feeding, protecting the mushroom until it releases spores. Psilocybin evolving independently in two distant mushroom families strengthens this defense idea. Convergent evolution implies strong adaptive value. Being eaten by insects is a major evolutionary pressure, and psilocybin looks like an effective solution. It's chemical warfare on the forest floor. From this view, the mushroom's magic is a targeted poison to manipulate predators, but the antefeedant hypothesis isn't proven yet. Many questions remain. Does it affect all insects or only some? Do real-world concentrations change insect behavior and survival? Some researchers suggest alternatives. Maybe psilocybin helps internal metabolism or helps compete with soil microbes. The full story is probably complex. Still, convergence makes the defense hypothesis the most plausible explanation so far. The revelation that nature has more than one recipe for psilocybin is not just a fascinating piece of evolutionary trivia. It has profound practical implications for biotechnology and for medicine. As interest in psilocybin as a therapeutic agent grows, so does the need to produce it in a pure, consistent, and cost-effective manner. Synthesizing complex organic molecules in a lab can be difficult and expensive, often requiring harsh chemicals and multiple steps. A more elegant solution is to harness nature's own machinery, enzymes. This approach, known as biosynthesis, uses biological systems to build molecules. The discovery of a whole new set of psilocybin-producing enzymes in inocybe mushrooms effectively doubles our biotechnological toolkit. Scientists now have two different enzymatic toolsets to work with. They can compare efficiency, stability, and specific functions. And they can compare enzymes from psilocybe and from inocybe. One set might be better for production in a yeast host, while the other might suit a bacterial host. The other set might be more robust or work faster under certain conditions. Having these options enables a mix-and-match approach, letting bioengineers pick the best enzyme for each step in a custom-designed production pathway. That could spark breakthroughs in manufacturing psilocybin for clinical use. A more efficient biosynthetic process could sharply lower production costs. That would make psilocybin-assisted therapy more accessible if it gains regulatory approval. It could also enable cleaner production methods, avoiding toxic solvents and reagents used in traditional chemical synthesis. Understanding these pathways also opens the door to creating novel, related compounds. By tweaking enzymes or tweaking pathways, scientists could produce analogs of psilocybin with different properties, maybe a shorter duration of action or fewer side effects. The branched pathway in inocybe is especially intriguing pharmaceutically. It provides enzymes to make psilocybin baeocystin and other related tryptamines. These minor alkaloids have long been a mystery. We know very little about their pharmacological effects. Now, with the enzymes identified, scientists can produce these compounds in pure form for the first time. That enables rigorous study to see if they contribute to therapeutic effects or if they have unique medicinal properties, opening new avenues for drug discovery. The story of psilocybin's dual invention is a perfect illustration of how science progresses. A new discovery answers old questions while simultaneously opening up a host of new ones. We now know that psilocybin was not a one-time fluke of evolution. Its independent creation in two separate fungal lineages confirms its deep biological importance. Yet, this confirmation only deepens the central mystery. What is that importance exactly? The antecedent hypothesis is a strong contender, but the case is far from closed. We stand at the edge of a new field of inquiry, looking into the complex chemical conversations that take place on the forest floor. The questions that arise from this discovery are as vast as the mycelial networks themselves. How many other times has nature invented psilocybin? Could there be a third or even a fourth undiscovered pathway in some obscure fungal species waiting to be found? And what about other complex natural products? This clear-cut case of molecular convergence prompts us to wonder how many other valuable compounds, antibiotics, anti-cancer agents, other medicines have also been independently invented by different branches of life. We may be surrounded by evolutionary echoes, similar solutions to universal problems, hidden in plain sight within the genomes of the organisms all around us. This discovery is also a profound lesson in humility. It reminds us that the fungal kingdom, so often overlooked, is a realm of immense biochemical sophistication. For every secret we uncover, 1,000 more remain veiled. These organisms have been experimenting with chemistry for over 1 billion years, and we are only just beginning to read their laboratory notebooks. The fact that they engineered a molecule that so profoundly interfaces with the human mind, and did so twice, invites us to look at the natural world with a renewed sense of awe. It suggests a deep, underlying connection between the chemistry of our own brains and the chemistry of the wider biosphere. In the end, the tale of psilocybin's two origins is a story of both wonder and utility. It provides practical tools that may help heal human minds, while at the same time presenting us with a beautiful intellectual puzzle. It pushes us to continue exploring, to keep asking questions, and to appreciate the intricate elegance of the evolutionary process. The mushrooms are not giving up their secrets easily, but they offer us tantalizing clues. They remind us that the greatest mysteries are not always out in the cosmos, but can be found right here on Earth, in the quiet, creative, and endlessly surprising world of fungi. Convergent evolution has written psilocybin's story twice, once in psilocybe and again in inocybe, with dissimilar enzymes and reactions converging on the same molecule. This rare molecular convergence deepens the mystery of why psilocybin and expands our toolkit to build it better and cleaner for medicine. Thank you for watching. Please subscribe, like, and share. Think about it. A new world awaits.